Good afternoon. This is Group 5 presenting Figure 6, Justin, Josh, and Mallory.
This is Justin. Normal autologous iPSC-based cell therapy avoids immune rejection since the immune system sees the transplanted tissue as itself. But when diseases affect cells that cannot be isolated, another cell therapy is needed. In Canavan disease, the cells are difficult to isolate and causes the loss of ASPA enzyme function, which normally breaks down N-acetylaspartate in oligodendrocytes in the brain. Without functional ASPA, NAA or N-acetylaspartate builds up in the brain, disrupting the oligodendrocytes' osmotic balance function. This causes water to enter the white matter of the brain, forming vacuoles and spongy degeneration.
Excuse me, but we talked about Part A. In Part A, the process begins with a simple skin biopsy taken from a patient with Canavan's disease. The fibroblasts from the biopsy are reprogrammed into induced pluripotent stem cells, also known as iPSC. Using GMP compliant and integrated free methods, the iPSCs still containing the patient's mutated ASPA gene, but they serve as a renewable source of cells for later correction. Before moving forward, the iPSC lines go through several quality control steps, such as checking their purity, karyotype, and expression of stem cell markers to make sure they are safe, stable, and suitable for gene editing. Once they meet all the criteria, the cells are cleared for the next stage of the process.
Part B focuses on repairing the genetic defect and creating the therapeutic cell type. The patient-derived iPSCs are genetically corrected either by CRISPR, TALEN, or by introducing a functional ASPA gene through lentiviral transduction. After correction, these iPSCs are differentiated into neural progenitor cells, NPCs, which are the cells needed for treating Canavan disease. Another round of quality control follows to confirm that the corrected cells show proper ASPA activity, have the right transgene copy number, are free of contamination, and no longer contain undifferentiated iPSCs that could form tumors. When all checks are passed, the corrected NPCs are considered ready for transplantation into the patient or for testing in preclinical animal models.
Experiments on mice with Canavan disease are used to test the effectiveness of this alternative cell therapy. Before transplant, the mice have myelin defects, motor dysfunction, and spongy degeneration, along with vacuolation. After transplantation of the WT, ASPA, and PCS into the corpus callosum, the subcortical region, and the brainstem via stereostatic injection, the mice had improved ASPA function or activity, decreased NAA levels, increased myelination, reduced vacuolation, and improved motor function.